a giant of the field, Patricia Demore, and she's my sister on the FASEP journal, so that's a good news as well. And I know Patricia for just a few years, I would say, right, Patricia?
Yeah,
we don't need to go into any more details, but clearly her contribution to field is just incredible. And for a while, I was thinking that maybe there were very few people who got a Kogan and Proctor Award, and Patricia is one of those. Very, 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 very few people. So she was not only a great scientist in her mid-career, but also she was a hope for the future, and that's why she got the Kogan Award. And indeed, this is the most challenging award, because then you have to prove yourself. Patricia did. So without any delay, let's just move on to our formal introduction.
Good morning, everyone. Thank you for your time and attention today. My name is Josiah Tow, and I'm one of the postdoctoral fellows in Dr. Andrew Brown's lab. It's my honor to introduce this guest speaker this morning, Dr. Patricia Demore. Dr. Demore is one of the pioneers in ophthalmic angiogenesis research who identified the infamous vascular endothelial growth factor in the eye. She was the first to demonstrate in vitro hypoxia-induced retinal pigmented epithelium VEGF, which led to further experiments that first correlated VEGF with in vivo ocular angiogenesis. Her work was foundational to the paradigm shift in using anti-VEGF therapies that we use today. She continues to push the boundaries of research by investigating the pathogenesis of dry AMD, as well as Endomusin's role in inflammation and angiogenesis. Dr. Demore completed her PhD in biology at Boston University, her postdoc at Wilmer Institute and Johns Hopkins University, and her MBA from Northeastern University. Although her first academic appointment was at Johns Hopkins, she ultimately accepted a faculty position at Harvard Medical School. Since starting at Harvard, she has held multiple assignments, some of them which are Schepens Professor of Ophthalmology and Pathology, Director of Research at the Schepens Eye Research Institute of Mass Eye and Ear, the Associate Chief of Basic and Translational Research at Mass Eye and Ear, and recently the Vice President of the American Society of Investigative Pathology. She has over 164 peer-reviewed publications and counting, 74 reviews, and is an co-editor of 4 books. Although she has won numerous awards, some of them being from Alcon and Arvo, one of them being also as a laureate of the Antonio Champalamo Vision Award, and lately the Kogan Award. She is very well decorated. Please join me in welcoming the esteemed Dr. Patricia Demore.
Thank you. Thank you very much. She has screen. We practiced this before, so we know it works. Okay, there we go. So I don't know, do you, if you want to, in this format, if you guys actually interrupt for questions, or do you want to wait to the end? Too complicated to interrupt for questions? Somebody would have to be looking at chat for me.
So we will do both. Those who cannot hold it will interrupt you. And anybody who has a question, just send me the chat that I have a question, and I will try to coordinate so we go chronologically as the questions are line.
So I actually, why is this not moving forward then? There we go. So this was the title, and I removed inflammation because it's 45-minute talk, and I'd like to leave room for questions. And also, the inflammation work has been published. I'm happy to give you a little recap of it with respect to this new molecule I'm going to talk about. But our newest work is in angiogenesis. We have a hope to get back to the inflammation part, but we're not there yet. So this is what I'm going to do. I'm going to give you some background on this molecule. In particular, for trainees on the call, I'm going to give you some idea about how we even came across this molecule. It was the result of what study section members like to call fishing expeditions. So just to say it works out sometime. Early evidence that we have a role for this molecule, and then where we're going with this now. So this is the end part of this is very much a work in progress. Interested to get some feedback, particularly on some of the mechanistic studies that we're doing to try to figure out how this works. This is the molecule. So it's a very small mucin by mucin standards. It's 150 fully glycosylated. And if you talk to the real mucin people who look at the front of the eye, the mucins they look at are in the millions. So this is quite small by their standards, but it is heavily O-glycosylated, some N-glycosylation I'll tell you about later. Has a small intracellular domain that has potential phosphorylation sites. And because of this heavy O-glycosylation, it's a very rigid structure. So it stands out from the plasma membrane. How did we find this out? Eric Ng, who loves me to show this picture because now he's a faculty at our place. We were very interested in trying to figure out at one point how tubes form. So how do you actually make a tube? What's the morphogenesis? What's involved in that? People knew lots about the cell biology part of it. We knew the cells migrated, proliferated. We knew they produced proteases, but we didn't really know how morphogenesis happened. So we happened to have, from work we were doing at the time, cystic embryonic stem cells. So I don't think people are doing this technique much anymore, but you can take an embryonic stem cell, these are mouse, you can force it to aggregate. We did that by putting maybe 2,000 cells in drops and then flipping the plate over, and then the drops all fall to the center of the cells all fall to the center of the drop and they aggregate, and then they grow to cysts or embroid structures that are really visible to the naked eye. You can then force them to sit and spread, and when you do that and stain for an endothelial cell marker, you can see that they're making all these tube-like structures, and you can see in cross-section that they actually have lumens. But people have used this model for lots of things. You can force it to produce cardiac tissue, and you can look in and this this little cystic structures are beating or contracting. So this was the model, and we knew that they could make tubes. So we took advantage of the fact that we had embryonic stem cells that were mouse embryonic stem cells that were VEGF null. So we made these little embroid bodies from wild type, and this is what I showed you before. This is just fluorescent stained. You can see the tube-like structures. But when we made these structures from VEGF null cells, we got endothelium, so you can see they're still staining, but they were not forming tubes. They were just making these little blobs. So we figured we had a model that we could use, and at the time we did, I think it was a microarray at the time. This is how long ago we found this molecule. We only recently revisited it. But we compared these two, and then when we looked at the top hits that were missing from the null, dramatically downregulated from the null embroid bodies versus the wild type, we came up with calvelin, which is a molecule that's been shown, even though I don't think the mechanism is well understood, in knockouts to have an effect on angiogenesis. We found Hay, which is a transcription factor downstream of Notch, and we didn't go for that because there's a lot of people working on Notch, but then we found Endomusin. And we sort of looked at it on and off over the years. We did develop a sort of interesting story with respect to inflammation, which, if somebody's interested, I can give you the Reader's Digest version of that later on. So it is an anti-adhesive molecule that is involved in vascular inflammation and in leukocytes sticking to the endothelium. So if you're interested, I could talk about that a bit. Has some interesting characteristics. This is a cross-section of a mouse pancreas, and what you can take home from this, you can look at this artery, and you can tell this is an artery because it has a medial layer, which means smooth muscle, and it also, upon fixation, if you don't perfuse fix, the luminal surface gets thrown into these waves, and that's because of the contraction of the vessel. And this is arterial endothelium negative, does not express, most arterial endothelium, never say never, does not express endomusin, but you can see the vein does, this collapsed vessel with very little medial layer, and also the capillaries. We've also shown lymphatics do not express it. So highly expressed by capillary and venous endothelium, but not at all by arterial. And that actually was part of the clue that sent us to looking at the adhesion part, because in the inflammation part, because what happens on many things, but what happens on the venous side of the tree that doesn't happen on the arterial side of the tree? One of those things is extravasation of inflammatory cells. So that was part of that. We also showed that it was luminally, so it's on the surface of the endothelium that's facing the lumen. This is human skin. And then we went back and we collagen stained it, so you can see that it's exactly on the lumen here. So it's very polarized in its expression. And then what is it a part of? It's actually a part of a structure, if you will, that those of us of a certain era will remember being referred to as the glycocalyx. It's sort of an old-fashioned term that's making a comeback because, as you'll hear, the components of the glycocalyx are biologically very relevant. So glycocalyx basically means coat. The calyx is the coat, and the glyco is the sugar. So it's basically a sugar coat. This is a picture. I think they stain these with Alcian blue, but you can see that these molecules, these glycocalyx molecules, rise, as I suggested to you before. They're very rigid. They rise from the surface of the endothelium. And I don't want to go into a lot of detail, but to suffice it to say that this structure, if you follow it throughout the vascular tree, varies a lot depending on where you look. So this is in vein. If you looked in artery, it's much shorter. There's been a lot of interest in this structure in terms of sepsis. But otherwise, there's not been a ton of research on, what the glycocalyx is really doing. It's mostly perceived to be an anti-adhesive structure. So, and I'm showing you here that the adhesion molecules that would be involved in inflammation are shorter than the glycocalyx, which turns out to be relevant in that story. So, in the field, it's known to have permeability and barrier functions because it's heavily charged, and in and of itself, it can exclude things from getting even to the junctions of the endothelium that would get them across. It's been shown to have mechanotransduction properties. It has anticoagulant properties. There's heparins as part of many of these glycoproteins, so that in itself is an anticoagulant, anti-inflammatory, as I've referred to. Also involved in organizing receptors, which may be relevant to the story that I'm going to tell you about today. It also communicates with neighboring cells. Growth factors can get onto it, chemokines, and when you hear of interactions between cells that people call juxtacrine, it's when neighboring cells communicate with one another. Many of those molecules are sequestered by the glycocalyx. So, and then what I referred to a moment ago is loss of the glycocalyx, or modification, has been associated with a lot of disease, particularly sepsis. If you search glycocalyx and disease, sepsis is a big one. It's more functional, really. I don't think people know mechanism that much there. There's recent discussions about glycocalyx changing in diabetes as well as atherosclerosis. So I think there's a big field to be investigated there. What are its characteristics? As I said, it's really negatively charged, so it can have an impact as a barrier. And what's in it? Hyaluronic acid, heparan sulfates, lots of plasma proteins, and then molecules like the musins that I talked about today. So to go back, I'll remind you again that the VEGF null cystic embryonic bodies don't make tubes. PCAM's another endothelial cell marker. Endomusin, as I've told you, is a marker for endothelium and culture. It stains all endothelium and culture, and we have an explanation for that, which I can tell you about. But if you make the EBs from VEGF null, you get these clumps, and they have very low, if any, expression of endomusin. So that validates the array studies that we did. So the hypothesis is very simple. Endomusin plays a role in vascular morphogenesis. So this was work that we did early on. This is looking at how the expression of endomusin in the developing retinal vasculature. So if you're not aware of this, the mouse is born without a retinal vasculature, and the full retinal vascularization happens in about the first 10 postnatal days. So if you look at a P3 mouse, if you looked earlier, there'd just be a tiny bit of vessel here. Isolectin B4 is an endothelial marker. Endomusin, I've told you, marks endothelium. So if you look at the P3, you have the retina in this case would probably be way out here. The vasculature forms up from the optic nerve outward in a centrifugal pattern. So you can see that early on, all of the endothelium stain with endomusin, meaning they're all expressing it, and you can see this total overlap. But what we also know about this is that this is a plexus. These have not really differentiated into arteries versus veins yet. Amazingly, just 4 days later, you can see the remodeling that's happened of the plexus. So you have true arteries and a true vein, okay? And interestingly, when this becomes an artery, you can see that the endomusin staining is gone. So it's totally missing from here. And we have evidence that at least some of that mechanism is that it's downregulated by shear stress. So we've shown that. So you can see the vein continues to express, but the artery does not. And the other interesting thing that comes from that observation is that if you look at the tip cells here, can I go back? Like if you're looking out here, you can see that there's very high endomusin expression out at the tips. It stays back here, but it's very high here. And actually in the adult, which I'm not showing any pictures of right now, endomusin expression remains, but it's downregulated. It's quite low compared to this. So we did a bunch of in vivo and in vitro studies that I'm going to, it's been published, though everybody has too much to read. Probably nobody's read it. I won't be offended. Don't tell me. I'm going to go through this and just give you the overview. I'm going to go rather quickly through this just because it's pretty straightforward. So what we wanted to do in tissue culture, oh no, this one's in vivo, right? I can't see it because my face is over. Okay, it's in vivo. So I'm going to first show you the in vivo work. And we are now making a knockout mouse. We have the mice and we're confirming, validating the knockout right now. But this was done several years ago where we didn't have yet a knockout mouse. So we used siRNA. As you'll see from other data that I'm going to show you, for some good reason, lucky for us, this molecule knocks down really efficiently with siRNA. So Cindy, who did this work, basically looked at what concentration of Si did we need and what's the time course. Now I need to point out that when we're injecting postnatal mice, you're injecting into an area, one area obviously, and you're probably only influencing a quarter to a third of the entire retina. When we analyze the knockdown, we're analyzing the whole retina. So you could have, and you'll see, total knockdown in one area and not much of an effect in the rest of it. So so we're doing the entirety of the retina. So you can see you get good knockdown with various concentrations, and you can see the time course of it. So we knew the dose.
Excuse me?
Yep.
This is Dorota. I maybe missed, and of course I can go back to paper, but if you could just say, how do you pack the siRNA before injection? Is it
lipofectomy? Or dermavectomy it was. I don't know. I'd have to go back and look because it's been a while ago, but it's in a lipid vehicle.
Thank you.
So you can see the reduction and you can see the time course. Gets pretty good knockdown, and then you get obviously a recovery. So the experiments were done in this time frame. And now it's not forwarding. There you go. So you can see and she would tattoo, with a little India ink, the area where she injected, so she knew the part of the retina to look at. And you can see if you look with the SI control, you have a good developing front. Remember, we're going to be, these are young pups that we're injecting. I think we end up doing P5s and this good endomusin expression. And then if you do the SI knockdown, you can see you knock down a good amount of the endomusin here. If I showed you the whole retina, you'd see that it wasn't knocked down in other parts. And then you can also already see, without me showing you the enlargement, the high mag that I'm going to show you, that this is looking reasonably impaired. And so if I get to a higher power, just better off to jump all the way over to here. But you can see the context of the whole retina. You can see that in the wild type, I hope you can see this, there are these very fine filopodia and lamellipodia, and that's believed to be those tip cells of the growing endothelium sort of reaching out to a gradient, which we know is VEGF in this case, because that's been well studied. But if you look down here, you'll see that those are very blunted. You don't see these lamella and filopodia, as if they are not sensing the VEGF, which I'm telegraphing the next part that I'm going to show you. And then we just quantify all that. So just because you need to quantify it. And again, this is significant, but not complete, because we counted the whole retina here and not just the quadrant that we influenced. This is just a picture to remind me to talk about where we're going with this. If you look, this is the oxygen-induced retinopathy model. Not sure if people are familiar with it. It's basically taking newborn pups with their the dam, putting them into high oxygen, and mimicking retinopathy of prematurity, so that these vessels are clumps of vessels that are growing out of the retina into the vitreous, and in some respects mimics, because it's hypoxia induced. And we initially were did a few experiments in this regard with siRNA, and we could knock down some of this, but we have the mice, so we figured it wasn't as clean. So we're going to be coming back with the knockout mice and doing obviously this in the CMV model. And I can tell you when we get to the end why that might be relevant clinically, or we hope that it will be useful translationally.
Very quick question, please.
Sure.
So this is a total knockout, right?
No knockouts yet. We are going to do a total knock. We have the knockout that we're producing.
Yeah.
The knockout that we're producing is floxed. So we flox the gene, and we're going to do, we're going through it now. We're going to do a total knockout, then we're going to do a endothelial cell specific knockout, and then we can do it conditionally. So this is just wild type, and the efforts we made here were just with the wild type mouse in the beginning.
So was injected with the siRNA?
No, we did. I'm not even showing that. I just put this in to bring up the topic. We did try that, and we did have an impact on that, but it's just not a very clean experiment. And when it became obvious that we had the floxed mice, we thought, don't do it this way anymore. The next seminar I give, or whenever we go to ARVO again, I'll hopefully be presenting the effect of the knockout on pathology. So this is just sort of a reminder for me to talk about that. We did see an effect, but it's just not clean enough to talk about. Make sense?
Thanks.
Okay.
Yes, thanks.
Okay. We also did it in tissue culture cells. And again, showing you, we did gain of function and loss of function. And this is just to show you get spectacular knockdown of this molecule. It's like 99%. And we also made an adenoviral vector where we could overexpress it because we wanted gain of function, loss of function. Now, the cells are human, and the vector is expressing mouse. So that's why you can see here that it looks like there's no endogenous. It's no endogenous because we're probing this with mouse and it's human cells. You can see what they have of their own to begin with right there. So it's significant overexpression. And not the most important experiment ever, I don't think, but if we were going to do one side, we decided to do the other. So I'm going to show you 3 sets of data. They all are very similar, so I'll spend time on the migration, but then we also do proliferation and tube formation, and the results are similar, so I'll go through those a little bit more quickly. There's many ways to measure migration in endothelium. This is what's called a wound healing assay. Very simple. You grow the cells to confluence, and then you take a pipette tip and you make a wound down the center of the monolayer, and then you let the cells migrate for X amount of time. I think these were either 8 hours or 16 hours. I'd have to go back. Might be overnight, 16 hours. And you can see, so the wound this marks the original wound. And then if you transfect them with control SI, you get this amount of recovery. So you get a reasonable amount of recovery. If you let it go another day, it would have fully recovered. If you knock out the endomusin, you can see you have far less wound healing recovery. And if you look here, this quantifies the reduction in migration that we got. good. So we did overexpression. I didn't think it was a necessary experiment, I guess, and I also thought we wouldn't be able to see an effect because my intuition was that the cells are already migrating the fastest they can. Like, why would they migrate faster? But in case you'll see, in fact, that this does increase it. So here's just the AdGFP control, and this is a level of recovery, and you can actually get a sense here that it's more recovery, and it's a statistically significant increase. So this is in response to VEGF, let me point out. The other one was in response to VEGF. I'm sorry I didn't say that on the last one, but all of these will be in response to VEGF. And then the same is true with proliferation. If you knock down endomusin, this is how cells proliferate in response to VEGF. If you take out endomusin, you get much reduced proliferation. Here's the control again. If you overexpress endomusin, you get statistically significant increase. And then finally, the same thing happens with tube formation. If you knock down endomusin, this is what the control looks like. If you use SI control, this is a positive code. This is VEGF-induced tube formation. But if you knock down the endomusin, you can see that the tubes are very fragmented here. And again, you can see the reduction. In fact, it reduces non-VEGF tube formation, just endogenous tube formation. But this is really what we're talking about here. And then this is even more dramatic, I think. These look so much more robust. This is overexpressing the control, GFP. This is what it looks like in response to VEGF. But if you overexpress the endomusin, you get these tubes that just, it's very empirical and subjective, but they just look more substantial. And these get measured by tube length, and you can see this increase with and without VEGF. So it looked like it was important for VEGF function. So we stopped and did a few structure function studies here just to see, and I'm going to go through these quickly because it tells us a bit, but I'd rather get to frankly, to the mechanistic studies because I think you'll all hopefully have some good suggestions there. So this is what the full length looks like. Remember, I told you that it has intracellular domain, we tagged them, transmembrane, and then there was this extracellular domain, and we truncated them based on where there were known predicted sites for glycosylation, because we wanted to get some insight into what was going on with glycosylation. I can't really figure out where to put this, these pictures of us so I can see people. There it is. This is just to show that they actually made it to the cell surface, because not useful. This is, remember I told you, a cell surface molecule, and we have other evidence done before this that I'll get to that shows it needs to be on the cell surface. So this is a biotinylation experiment just to show that all these mutants made it to the surface. I should say that we made two other ones that would have been interesting but didn't make it to the surface or didn't get expressed. One that had no intracellular domain at all, and one that was just intracellular domain and transmembrane domain, but no extracellular domain. And those did not express well, so we're trying to figure out what we can do instead. And then this is details about how we did the assay, which I'm going to stay away from for now. If you're interested, I can explain it. But basically, the important thing is we knocked down endogenous endomusin, because we know endogenous endomusin is essential for VEGF stimulated functions. And then we added these back, and the simple question is, which ones can rescue the absence of endogenous? So that's all we're asking for. And the take home is over here, where you can basically see that the smallest truncation mutant here is insufficient to rescue VEGF-induced migration when you take out endomusin. So does that make sense? So this is the intact system where you have cells with their own endogenous endomusin, and you add VEGF, you get stimulation. This is what I showed you before. If you take away endomusin, so all of these have endomusin gone. If you take away endomusin here, there's no stimulation with VEGF anymore. all of these mutants are capable of rescuing to here, except the smallest one. And that's what the data look like right there. We also wanted to see, I'll show you later on that we have evidence that the Endomusin actually binds to VEGFR2 and mediates internalization. And now that I think of it, I should probably have put the structure function stuff after these data. Haven't talked about this a lot yet, but simply to say that these are immunoprecipitations that we're doing, and you can see that they bring down VEGFR2. So you bring down if you blot with VEGFR2, you bring down Endomusin. And we also have shown that you get internalization. I'll show you those data much more convincing fashion in a minute. So this is what internalization looks like, and this is simple. We've shown internalization many ways. This is cell surface fraction. The way I'm showing it here is that when you add VEGF to a cell, it binds to R2. R2 gets internalized, and it's missing from the cell surface. It's gone, right, because it got internalized. So if you do biotinylation studies, you will see that, just go here, you add in the full length, you add VEGF, and you come back and look for R2, there's less R2. There's less R2 with this mutant, less R2 with this mutant, but you can see that with this small mutant, VEGF-R2 is not getting internalized because there's the same amount left on the cell surface as there was when you started. And I'll show you some convincing immunofluorescent pictures of that right now. So this work was done by Michel LeBlanc. This is just, I have co-localized in quotes here because everyone in the audience will tell you that light microscopy is not going to really tell you about co-localization of molecules. The IP data that I'll show you shortly is what you need for co-localization. But it does show that they're both on the cell surface. So you're seeing Endomusin and VEGFR2, and when you see this white, you can see that they're basically on the cell surface and generally in the same place, which is not surprising. They're both cell surface molecules. The real key was to find out if there was an interaction. Why does it keep jumping? The interesting thing here is twofold. Yes, you get an interaction of VEGF with VEGFR2, excuse me, let me back up. You get an interaction of Endomusin with R2, but interestingly, and not what we expected, it's independent of the presence of VEGF. So I think if we were thinking about this earlier, we expected that Endomusin was doing something to present VEGF-R2 or to stabilize VEGF-R2 VEGF interaction. So we thought, we anticipated that this would look different in the presence and absence of VEGF. But you can see that it's not. You have, when you're immunoblotting for VEGF and Endomusin, you're seeing the same. So there's no dependence of this interaction on the presence of VEGF itself. So we wanted to, this is also another topic that I misguessed in the beginning. So I was pretty sure, based on what we were seeing, that if VEGF, that VEGF would get internalized, but that the Endomusin would do something downstream to interfere with the signaling. So remember, the question we're asking now is, just to step back and look at the big picture, we have a mucin that's on the cell surface, and if we take it away, we're no longer getting VEGFR2 signaling. We just block VEGFR2 signaling. So what's the mechanism of what's going on there? So first we asked, did Endomusin influence VEGF phosphorylation? And I'm not showing those data. I can tell you right now that if we take away Endomusin, we still get VEGFR2 phosphorylation. So it's not the phosphorylation step that's being influenced. So to go downstream for that, what's the next thing that happens? Well, there's certainly a lot of adaptive proteins that get assembled, and we'll talk about those shortly. But VEGF-R2 internalization is important, and it's important because it turns out that at least for VEGF-R2, you can have phosphorylation, but the signaling continues in the cell after the phosphorylated R2 is internalized. So to get continued signaling, the R2 actually has to be internalized. So this is where we get to see, is that what's being influenced? In the assay that that Michelle set up for this was to incubate the cells with anti-R2. Okay, so they're all labeled now. Then incubate them with VEGF for 30 minutes, and this is in the presence of an inhibitor of recycling, because you don't want the receptor to go back and recycle. We just want to freeze it in space. So let the receptor internalize, and then come back and add a fluorophore, so that you can see what's going on. And then the low washes and the permeabilization take off everything on the cell surface. So now you're only looking at what happens when it's internalized. So not surprisingly, this is what it looks like just uncontrolled. So you have VEGF, and you're looking for what's happening right here. And you can see that the VEGF, this is staining for R2. So you can see the R2 sort of all over the place. If you add VEGF, though, you can see a distinct internalization. It's all sort of perinuclear right now. And that's not new. That's what you'd expect. This is after 30 minutes, and you can quantify that. Surprisingly, for me, though, what you see, again, this is back to the control. You're adding VEGF. This is with the control SI, so this is normal cells. If you take away the Endomusin and add VEGF, you don't get the internalization. So you can see that there's not much difference here. Now, granted, and I'm going to show you more biochemical proof for that. This is convincing, but it is immunofluorescence. And we could quantify that also. You can see this is this internalization, and then you take away Endomusin, and the receptor is no longer internalized. That's the comparison there. We also showed that it remained on the cell surface. You can show that by immunofluorescence, not that convincing, because again, immunofluorescence is a little difficult. But you can see here that if you add VEGF in the presence of the control, the staining is no longer cell surface mediated and is punctate throughout the cell. But if you go back here where you've removed the Endomusin, you can see how it's very much on the cell surface, again, staining for R2. The most, I think, convincing is the showing what happens in a biotinylation system. So this is looking at VEGF-R2 on the cell surface, bringing when you biotinylate everything on the cell surface. And you can see if you add VEGF when Endomusin's still around, a good percentage of the R2 disappears from the cell surface. But if you take out Endomusin, there's no difference. There's very little difference, no difference. You get no internalization. And that's quantified here again. So something about the absence of VEGF-R2, something about the absence of Endomusin on the cell surface prevents VEGF-R2 from becoming internalized. And even though it's phosphorylated, because R2 requires continuous intracellular signaling, you're not getting the biology on the end. So phosphorylation, but still no biology on the other end. And then this is a similar experiment, but this is to actually show that the Endomusin doesn't get internalized. So this is not knocking down Endomusin. This is just asking, when you internalize R2 normally, in response to the addition of VEGF, does Endomusin go with it? And my prediction was going to be, yes, it does. And the answer is, no, it doesn't. So this is it. Immunofluorescent, but again, this is the more important one. This is biotinylating the cell surface, and you see you have Endomusin, you add VEGF. We know R2 would go in, but the Endomusin is staying on the cell surface. So they have the dilemma here as we think forward, because I don't have an answer to this, is that you have a molecule that remains on the cell surface, has some kind of an interaction with VEGF-R2, because I've showed you that by immunoprecipitation, and is necessary for the R2 to get internalized. So this is the mystery that we're at right now. I'm going to summarize this and then talk a bit just about what we're thinking of in terms of next steps and where we've gone with some of this. So I've showed you that Endomusin, which is the cell surface molecule, is necessary for VEGF-induced functions. We've done this in tissue culture, and we've done it in vivo. We've shown that Endomusin binds to R2, and I didn't show you the data that the internalization that we're measuring is clathrin mediating. So you can internalize either by macropinocytosis or by clathrin, and most of VEGF-R2 is clathrin mediated. So this obviously gives us a clue about where we're going to go next. Endomusin doesn't get internalized with the R2, surprising to me. And then structurally, you need a minimum of 121 amino acids for the R2 function, for binding and for internalization. Things we don't know about the structure is we don't know if phosphorylation happens. We've made many efforts to see if we can pick up phosphorylation of the Endomusin under any circumstances, and we haven't seen that. So we're now making mutants where we're mutagenizing the phosphorylation sites to see if that affects the biology. That's one way to go, and that's how we're approaching that right now. A general conclusion is just that the glycocalyx is very understudied, and I think an underappreciated component that appears to contribute to a wide range of the functions. From a potentially translational viewpoint, Endomusin's interesting because it's cell type specific. So if you think about the widespread use of anti-VEGF right now in a variety of retinal pathologies, diabetic macular edema, AMD, central retinal vein occlusion, even ROP, that VEGF has access to all of the cells, anti-VEGF has access to all of the cells in the retina that express VEGF-R2, which is actually a lot of cells. So astrocytes, Mueller cells, photoreceptor cells, RPE, all of those cells express VEGF-R2 and to some extent depend on VEGF-R2 for their survival. Certainly not the only survival factor, I'm not trying to suggest that, but they do, you can demonstrate in culture and in vivo that if you take away VEGF, you can have an effect on those cells. So having a molecule that can block VEGF signaling that's endothelial cell specific might be an improvement. And that's what we're thinking of on one side of it. On the other side of it, there's a number of people that don't respond to anti-VEGF. Nobody knows why. There's tons of papers about polymorphisms. Nobody really knows what's going on there. Some people think it's yet another factor. I tend to not believe that. I think it may be that in some cases, the VEGF that's mediating the pathology is not accessible, could be sequestered on heparan sulfate or on a membrane or anywhere. And there's a possibility that by targeting the receptor instead of the ligand, we might have a better anti-VEGF effect. So those are the two thought processes about how this might potentially be some improvement or alternative or combination with anti-VEGF. So I just want to credit the people who are doing this. I'm collaborating with Eric Yang and Magali Sanjanez, who are both PIs. They're working on this with me. Ping is the person who did the structure function studies. Not pictured here is Michelle, who did the early mechanistic, and Cindy, who did the first descriptive work. And Isai is working on another matrix component that you probably have heard of, galactin-3, that looks like it may also play a role. And the work is funded by NIH, and we had a grant from the Grimstraw Foundation. And Dietmar Westweber was very generous in providing us from some anti-sera. So stop there and be happy to entertain questions.
Patricia, thank you so much. I'm sure there's quite excitement. Let's start with two questions from John Hong.
So should I?
Hello. I was wondering, is the effect by Endomusin on neovascularization by VEGF signaling mainly from Endomusin, or do you suspect that there might be some other glycoproteins and extracellular matrix proteins that might also contribute?
Yeah, I think, the whole idea of specificity is an important one. We just are finishing data right now, though, convincing data to me, that show that, for instance, Endomusin is not necessary for FGF-induced angiogenesis or stimulation. And FGF is a great angiogenic factor. And if you knock down Endomusin, you have no effect on that. Are there other molecules? We tried galactin-3. Galactin-3 is not necessary for VEGF-induced functions or for the Endomusin part. We haven't, systematically looked at every glycoprotein out there. Some of them will be hard to knock out. Like if you knock out heparan sulfate, you're going to have an effect on many things. But I think it's a good question. Is there redundancy in the system, and how could we look at that? So,
my second question was, looking at your internalization studies, I was wondering what the interaction of Endomusin with VEGF-R2 versus phosphorylated VEGF-R2 was. Maybe there's some difference in interaction with phosphorylated and non-phosphorylated.
Yeah, we didn't see, though. We did the interaction plus and minus VEGF, which would therefore mean plus and minus phosphorylation, and we didn't see any effect there. Would have been nice if we did. So it looks like that's a little bit of the confusion. It looks like it's always there. Phosphorylation doesn't look like it has an effect on the interaction, at least from that IP, which we haven't, for instance, looked at affinity. We haven't done anything quantitative like that, which is probably necessary to address your question.
We have a large number of questions, so let's move to Sri Ganesh.
Somebody muted?
Okay, looks like he's not there, so let's move to Jim Howe.
Yeah, thanks. So I have a couple of questions. First, could Endomusin be playing a role in keeping veins from collapsing when they're low filled by virtue of the charge?
You know what? That's so funny that you say that, because way early on when we tried to look at this, that was one of our actual hypotheses, is that, and we don't know that yet. That's actually something we'll be able to get to see in the knockout animals. When we get that, but because the vessels collapse so easily, and it is sort of a repulsive factor, it's a possibility. And we did try to do that in tissue culture unsuccessfully at one point. So we'll have to see. But I can see how you would think that just from looking at the in vitro pictures from the cystic embryoid bodies. Yeah, it's a good idea, and we'll definitely. Adding it to our list of phenotypes to look for.
And does the charge change in the delta 21-161?
Oh, that's a good question. It has to, because we've cut off a lot of the. I think we have a couple of O glycosylations left. So as we clip it down further, we're cutting off more of the O and N glycosylation. So it's got to. We haven't measured the charge there. We could probably do an isoelectric gel or something like that and see, but we haven't done that. But I would predict that it would, because it's so much less glycosylated.
Yeah, and I have one last question, and that is it possible that that the Endomusin is passing the VEGF receptor off to something else?
Yeah, I mean, we're going to have to look at that. So we just had this big discussion yesterday about what our next steps are going to be. So this is a clathrin mediated phenomenon, and we looked in the vis-a-vis the R2 literature. There's nothing out there that suggests something like that is happening. So we're going to have to sort of reinvent ourselves to now be learning about endocytosis. But, there's a lot of known molecules. There's a bunch of adapter molecules that are involved downstream. But I think we have to go way upstream. So the question is, does the membrane even invaginate? We're going to end up actually with plan to do EM. The only way to look at that is by EM. So do you get the beginning of the clathrin-coated pits? And that we're going to have to look at in Endomusin null cells to see. So we're just beginning those studies now. So I'm totally open to any good suggestions.
Thank you very much.
Thank you.
Let's just move to in the sequence of Dorota, Aisha, and Susie.
Thank you. That was pretty amazing talk and lots of things to think about. But my question is a little bit on the angle of aging. So ECM really changes in aging. Do you know anything what's going on with Endomusin there?
No, we don't. But, I think part of the reason people haven't looked at glycocalyx much is because it's all glycobiology. And glycobiology is really challenging to look at. It's not like you run a Western blot, are you? Yeah. So where, for instance, I do believe that will be the case because, first of all, everything changes with aging, but also that people are seeing, have been able to measure changes in basement membrane, at least and glycocalyx with disease processes. The question is, how do you do that in a systematic way where you're not just doing an EM that shows suddenly that it's flatter, which is basically what happens in the sepsis literature. It's interesting to know that. But so, for instance, for the in the inflammation project that we did, we found out that when you treat the cells with a cytokine like TNF, or all of the endomusin gets clipped off. It just shears off the surface, and that's a twofold, at least, quote unquote purpose of that. One is you've gotten rid of the anti-adhesive thing that keeps the leukocytes from sticking normally. But the other thing you do is you clean the surface for the adhesion molecules to be the most exposed. So I think we're going to definitely see differences. I think for us, the challenge is going to be how do you do that in some way that you can quantify. And,
so this is really intriguing because my next question was about the response to stress, and you partially responded that. So, but potentially you can see the cleaved molecule accumulation with any stress or so on. But so nothing happens on the transcriptional level, or
no, not much on the transcriptional. Well, the clipping from the cell surface is immediate. Transcriptionally, you can look later and maybe see a little suppression, but what you do is you can do a cell surface fractionation, and you just see a little piece of the transmembrane and intracellular domains left. It gets clipped right off. Interestingly, it's hard to find the extracellular domain. I don't know if it gets chewed up or proteolyzed. There's a lot of proteases around when you're hitting cells with TNF, so my sense is it might be proteolyzed. But it's known for other glycocalyx molecules that they're shed during. And so the other question that we haven't gone to yet is when we do that, can the cells no longer respond to VEGF? Because the prediction would be they couldn't respond to VEGF at that point. But that gets very messy, so we haven't done that yet. Or we haven't done it well. We've probably tried it once, but we haven't done it.
Yeah, very interesting. Thank you.
Thank you.
Hi, my name is Aisha.
Hello.
I have a few questions. Hi. So, first thing, is endomusin is altered in patients that show new vascularization or permeability?
Yeah,
Do we know?
nobody has looked really. I saw a paper recently where somebody suggested that there were reduced levels in diabetes. I think it was actually an Endomusin paper. It's a descriptive paper, but it's still very interesting. So actually, it's a good point. My question has been, I know if I hit the cells with TNF or interferon, I clip it. But what happens in the kind of inflammation that we're all interested in these chronic disease processes, which whatever you call it, para-inflammation or chronic inflammation, are the low levels of, say, cytokines that are present then going to do have the same kind of action on the glycocalyx as it would in like acute inflammation like sepsis, and we haven't been able to document that yet. We haven't, you'd have to be able to get human tissues, and then I don't think immunohistochemistry is going to be sufficient to quantify. You're going to have to be able to do Western blots or something like that to see if it's changed. But I think it's a good question, and it's one that sort of ties the inflammation story to the angiogenesis story. Is this going to be vulnerable to chronic inflammation? We don't know the answer to that, but it's a good question.
The other one is because VEGF receptor 2 interaction with the endomusin need VEGF.
No, it didn't.
To show the effect at the other end.
No, it didn't need VEGF. The interaction didn't need it. You need to get the biology, obviously, but we could immunoprecipitate.
No, internalization, I mean. Internalization, right?
Oh, yeah, definitely.
It needs internalization.
Yeah. Yes, it does, yeah.
So it could be that somehow VEGF is changing the conformation of endomusin.
So VEGF changes the conformation of, I don't know about that, because it's a pretty boring molecule. It's a stick that's heavily glycosylated, and I'm not really sure where that would get us, but it's possible. It's not a low-hanging fruit, let's put it that way. That's not the easiest thing to look for. But, we are stuck at, what about the Endomusin allows the R2 to get into a clathrin-coated pit and get internalized. And, anybody on this call has good questions. I have good questions. If you have a good answer, I'm interested to hear it.
And what is the role of endomusin in inflammation in general?
It actually reduces leukocyte adhesion to the endothelium under resting conditions. So if you take a normal endothelial cell and in the inflammation field, people use umbilical vein cells a lot, and you put, say, neutrophils on that, they don't stick. If you knock down endomusin, you get sticking, and it turns out that there's a good amount of constitutively expressed ICAM. So the adhesion molecules that we all think about that get upregulated by, cytokines, that's definitely one class, but all endothelium have low levels of ICAM there already, but it's apparently blocked by something like endomusin. So under normal circumstances where you don't want inflammation, the leukocytes can go by and not stick to the endothelium. But if you take the endomusin away, you get a good amount of sticking.
All right, let's move on to Susie and Andrew.
Hi, I'm Susie. I just have a quick question. I might have missed it during your presentation, but you showed an interaction between endomusin and VEGFR2 through fluorescence and co-immunoprecipitation. Was it done in tissue culture, or was it done with the actual mouse tissue?
No, tissue culture. We haven't looked yet to try to get that interaction in vivo, but that's always a good question. They weren't overexpression systems, which I think is important. If you do overexpression and you see interaction, there's reason to be a little suspicious. So they weren't overexpression systems, but we have not done it from tissue in vivo, and that's a good point. Much more convincing if you see it in vivo.
Thank you.
I have a quick question. This is regarding the glycosylation of endomycin.
Yep.
Have you tried an experiment where you treat endothelial cells with tunicamycin to inhibit protein glycosylation and to see if that inhibits VEGF uptake?
No, what we did though was even cleaner. We made I didn't put this in because then it gets down a rabbit hole. We made a mutant of. We took the smallest truncation mutant that still worked because it was easier to do this, and then we had a version of that made where all the old glycosylation sites were mutagenized so they couldn't get glycosylated. Because we figured that way we'll start grossly, get rid of all the old glycosylation, and then we could add them back and see how it worked. It had no effect. So we got rid of all the old glycosylation, and it had no effect. So the old glycosylation is not necessary.
But the protein is also N-glycosylated, right?
Yeah, and we're doing that now. We did a pharmacologic inhibitor of N-glycosylation, and it looked like it had an effect, but that's not as convincing. Mutagenizing those, and they only have like 4 or 6. The truncation mutant that we were working with only has like 4 N-glycosylation sites. So I thought it had no effect. I didn't think it was going to matter at all.
Was that on a null background?
Say that again.
Was that on a null background? The expression of the.
Yeah, we knocked down endogenous and then put back in the mutant. Yeah.
Knocked down.
Say it again.
Was that a knockout or a knockdown? The background.
No, we knocked down the endogenous, which, as I showed you, is pretty convincing. Yeah, maybe there's a couple of percent left, but it's enough to get rid of the biology, at least in terms of studies.
Just the last comment is all. It may be very interesting to look at what happens to this protein in congenital disorders of glycosylation, specifically the ones that affect O glycosylation.
Okay. That's a good idea.
Thanks. Very nice talk, by the way.
Thank you. Thanks for your attention. Anything else?
Okay, we have more questions. So the next would be Andrew and then Patricia Becerra.
Morning. Thank you for your really enjoyable presentation. I think that Sriganesh kind of came up with some of the questions that I had. As a clinician, I'm always thinking about how can we start to apply these kind of principles to patients, and of course you showed the ROP style model in a mouse. So I was wondering, are there pharmacological techniques for disrupting the interaction between Endomusin and VEGF receptor 2? I think that Sriganesh was kind of talking about tunicamycin inhibiting the certain components, but maybe an alternative would be if you disrupt VEGF receptor 2 recycling with primaquine in that model. I wonder if that would have value in ROP. In ROP, we struggle so much with the idea of putting anti-VEGF into a baby with so much VEGF-dependent activity and development. So I'm probably a little bit of a naive question, but I'm wondering.
Yeah, I think that would be too non-specific because you probably, almost certainly in such a developmental system, you have a lot of receptor activity going on, so you wouldn't want to block it all. My understanding in the ROP, this may be you may be doing this already, is that they're using, there's been a trial recently where they, if I'm correct, tested 1/100th of the dose they were using before, and that was sufficient. And I think it requires, I was never a fan of, obviously, anti-VEGF in an ROP model just because I thought it was a disaster waiting to happen. But the trial, I think, used 1/100th of the dose, and it only requires 1 injection. Still don't know if there's going to be anything downstream of that, but untreated, if it's untreated, it could lead to blindness. So my understanding is that is 1/100th of the dose is working well. If that's the case, then it's better than what was used in the past, which was like cryo and laser and stuff like that. We're thinking of trying to interfere with the interaction with an antibody. That was one idea. So we made one effort that didn't work at making monoclonals that could interfere with the interaction. that would obviously be way more specific. We may also just be able to find a peptide at some point. That might be even cleaner. But we're not there yet. And assaying for antibodies by immunoprecipitation is painful. So, but I think those are good questions. Of like, if we do think this is a good target, how are we going to target it? And antibodies may be good because they're tried and true. They stay around long enough. You can humanize them. There's enough technology there that may be better. Peptides would get cleared. How would you keep them around? So I think an antibody approach is probably a good one. So we're probably going to go back and try that again, another round of monoclonals, and see what we get.
Thank you.
Patricia, are you there?
I am. Me? Or the other Patricia?
No, the other one.
Yeah, I don't know. She might have had to leave. Patricia, she wanted to know, is there evidence that glycosamine glycans upregulate VEGF-R2 axis? Let's see. Upregulate the VEGF-R2. Well, it's just an empirical observation. Every time we knock down Endomusin, we get elevated VEGF-R2. Every time, it's like the most reproducible thing. So there's some feedback thing going on there. It's not a SI problem because we don't get it with the controls. So knocking down that particular glycosaminoglycan does upregulate just the expression of R2. That's the best I can answer on that. And how many O glyco groups were there from the biggest to the smallest? I don't remember. 16 maybe? There were a lot. It's very heavily glycosylated. We picked I can go back and look if somebody's really interested. I don't remember. I'd say it was 16. We picked the ones that were most likely to be O-glycosylation sites and got rid of those. And
all right, David. I'm sorry, Emma, and then David, and then Avery.
Okay.
Hi, Dr. Diamore. My name is Emma. I'm Dr. Karen postdoc fellow. I have a couple of questions. Very nice talk, by the way. It's Endomusin works by itself. Have you seen if it forms any type of rafts in the cell membrane that might facilitate the access to the VEGF receptor?
That's a great question. And we don't know that yet. We haven't been able to. It doesn't interact with itself that we can find out. But then there are other molecules out there, like these galactans, that are thought to not cross-link, but interact with extracellular molecules and help them aggregate. So is it an aggregation thing? Do we need to aggregate? Is Endomusin, one possibility is Endomusin's involved in aggregating the R2, which is a step that's necessary to get internalization, and we don't know that yet. It's a very good point. It's a good suggestion, I think, and a good option.
Okay. My second question is, in your wound assay, you show how knocking down Endomusin, there is no migration of the cells. Have you looked to see if there's any relationship of Endomusin and the Wnt signaling pathway?
No, we have not. But there is interaction between the VEGF pathway and the Wnt pathway.
Right.
So I would not be surprised if Wnt was involved in the assemblies. But we've stayed away from looking at the downstream because we do get off in that abyss sometimes. We're like, let's look downstream. No, people already know how VEGF signals, and clearly if we're not getting signaling, we're not going to have downstream signaling late. Oh, that's an interesting thing too. We've done a bunch of phosphoblots where we've looked at time courses of known signaling molecules. In the early signaling, like ERK and AKT and that, up to 30 minutes is normal. And then when you get to 16 minutes, it's very reduced. So it means that the sustained, that's a better word, the sustained signaling that you need once the receptor gets internalized, it's what's missing. So we haven't followed that though, because I think that's now makes sense, it's consistent, but it's not going to solve the question of what's it doing. So every time we have discussions and people want to do that, I keep pushing them upstream to try to figure out what's happening at the membrane.
Right. And my third and last question. you have been interrogating human retinal endothelial cells. Have you been done this type of experiments in other types of endothelial cells? Endothelial cells that renal endothelial cells, or because I think there's an heterogeneity on the endothelial cells depending where they are present.
So we've used umbilical vein cells because we did the inflammation work on that. My bias about the heterogeneity is you lose that pretty quickly once you're in culture. So if it's almost certain. So the arterial cells, aortic cells, once you put them in culture, they're missing all their environmental clues, and they express endomusin. Why? Because the shear stress is gone. Brain endothelium, which we've cultured many times, which has spectacular trans-endothelial resistance, one passage and that's gone. So, if you wanted to do that, you'd have to use, primaries. And that would be interesting to do for sure. But, looking at cells that are passaged in culture, I'm always surprised. You might have some epigenetic changes that you can sustain, but I think we're going to find out in vivo when we get to start looking at knockouts or conditional knockouts if there are tissue-specific differences in the function of the endomusin.
All right, thank you.
Thank you for the nice questions.
Why so many O-glycans?
I think the O-glyco. Well, I would have thought before that they were entirely necessary for keeping the molecule extended enough so that it could do its interaction. But as I've told you, we took them out and it didn't appear to have an effect. So I now don't know. It may be in another function. So it may be that if we used the O-glycosylation deficient version in the adhesion assay for the inflammation story, that it would not do as well there because maybe it's a little more floppy. But that's entirely a guess.
Is it possible they just stabilize the protein against the extracellular matrix proteases and has no really functional role?
Yeah, it may, because that's a good point, because the studies that we've done have all been in tissue culture. We haven't tried to knock out O-glycosylation in vivo. I think that would be really hard. So many molecules are O-glycosylated that it would be hard to even interpret the results. But we were shocked at not having a result. But it's a good point. We have not gone back and used those mutants in the adhesion assay, and that may be different. It may be that it's less rigid and therefore not as good as protecting the cells from inflammation or leukocyte adhesion, and we haven't done that.
All right, the time is late. Let's just have a last two questions. One will go to Avery, and then we will finish with Dr. Gong.
Hey, Dr. Diamori, it was a really, really nice talk. Considering that the O-glycosylation doesn't really impact endocytosis of the VEGFR2, I was wondering if maybe if you just added the extracellular domain after knocking down the entire protein, do you see a similar response?
Yeah, that's a great question. We haven't done that because when we made the construct, we didn't make it the way you're suggesting. We should make it without the transmembrane domain, express it, collect it so we can add it back. We didn't do that. We made it with the transmembrane because we had a different question at the time. We were like, oh, do we need the intracellular domain? But that's a great idea, is to make that and see, does it really even need to be tethered to the membrane to work? I'm writing down your idea.
Well, I figured that something has to recruit the adaptive protein complex. Yeah. And considering that endomusin doesn't get endocytosed with it, then there might be another intracellular pathway that's recruiting the complex. So maybe, I don't know, there's a lot of questions that can stem from the exact mechanism. So
right.
It can be also an excellent inhibitor, just in case.
Right.
That's right. That would be So at least two reasons to make it. Good idea. Is there another question?
Hey, Patricia, it's Shahar Gong from UC Berkeley. I was just wondering endomusin's function in the mature vasculature. So So the interaction between the VEGF R2, is that suggesting that R2 regulate different between artery versus the veins?
I heard that you were saying in the mature. What was the last sentence? I missed the last sentence.
So the interaction between Endomusin with the receptor R2. So does that interaction reflect the difference between the R2 function in the artery versus the veins?
That's a very good question because let me think through this now. That's interesting because it gets very complicated. It's too long for this. So the question is, because you see, you don't see endomusin in arteries. Do arteries respond differently to VEGF than veins do? Well, you know what? There was the whole idea that floated around for a while. I don't think it's been totally worked out. That some cells can make VEGF in an integrin fashion, and it signals integrin internally, so that you don't even need to internalize it. That the receptor is in the cell, and that the cell is making VEGF in the cell, and that they're meeting and doing this signaling there. That's a good point. That would be a good thing for arteries because they're not making the endomusin. So that would be a good explanation for why you'd have to have an intracrine system. The other reason is you can't have arterial endothelium making VEGF. The flow is too great. It would not work. It would just get floated away. Whereas veins, you can imagine, plus the fact that they have a glycocalyx might trap the VEGF, so it could work more locally. That's a really good question. Yeah.
So to follow up, you mentioned it also expressing capillaries. Do you see the sharp boundary disappears in terms of distribution of the protein?
Yeah, that's a good question. We have not looked, but we could go back and look easily at those flat mounts in the retina. We should. Does it cut right off? Or not? And we did the flow studies that we did were not ultra precise. We don't have equipment that allows us to manipulate the shear stress, but we did show using arterial endothelium that if we subjected them to shear stress, VEGF did get, endomusin did get downregulated, and we could show that it was by the classic KL4 mechanism. But that's a really, I don't know if it's a gradual or a sharp cut. We should look at that. That's a really good point. And we have the wherewithal. We can look in the retina. So, thank you.
Right, guys, thank you so much. We had a wonderful presentation, great questions, stimulating conversation. Now Patricia knows what to do for the next 5 years.
Yeah, thank you all.
Thank you very much. Bye-bye then.
Bye, everybody. Thank you.
Bye, thank you.
Thank you. Yes, thank you.